All right, now we're getting into the molecular genetics portion of the course. This will be the majority of the rest of the course. And here's an outline for that. Today we'll, in this lecture, discuss DNA chemistry and structure, chapter 10, then DNA replication, then transcription in RNA, genetic code translation, mitochondrial and chloroplast DNA, how genes are regulated, transposable elements and mutations, genomics, and lastly, epigenetics. And usually we have our second exam after chapter 10 or 12, so that will be coming up again within another week or so, our second exam. Okay, an outline for chapter 10 then. Discuss the central dogma of molecular biology, chemical structure of DNA and RNA, prokaryotic and viral genomes, and that will do it. So here's our central dogma of molecular biology. This is how information is stored and flows in molecules. So of course, DNA contains information for the phenotype. The circular arrow refers to DNA replication. First step in gene expression is transcription, where DNA serves as a template for the synthesis of RNA. And then the second step is where RNA serves as a synthesis, as a template for the synthesis of polypeptide chains or proteins. Some retroviruses are capable of reverse transcription. Some RNA viruses can replicate their own RNA genomes. And here's another way of looking at the central dogma in a little more, I guess, complex way. So, characteristics of the genetic material: it must contain complex information, must replicate faithfully, and it must encode the phenotype. And DNA fits all of these. Laying down the freeze-dried corpse there is Otzi the Iceman, who was killed 5,300 years ago in the European Alps. And they were able to successfully isolate mitochondrial DNA from him. Mitochondrial DNA, of course, is the circular DNA molecule found in the mitochondria of our cells. And it's been worked with a lot, particularly in animals. So, X-ray crystallography is a technique that showed that DNA is a helical molecule with bases stacked perpendicular to the long axis of the molecule. And the woman that did this work is Rosalind Franklin, and her boss, Maurice Wilkins, ended up sharing the Nobel Prize for this work. Talk about that more later. There's Rosalind, and she died at age 38 of cancer. X-ray crystallography, of course, uses X-rays, and she may have died from cancer due to too much X-ray exposure. This shows that back in 1928, here in the top center, a fellow named Griffith figured out that something called transformation occurs. And then in the 1940s, Avery, McLeod, and McCarty figured out that DNA was the transforming principle. Then Hershey and Chase in their experiments learned that DNA was the genetic material. And then in 1953, Watson and Crick published their paper on the structure of DNA. X-ray crystallography works this way. You shine X-rays through a crystal of some kind of substance, and it gives you a diffraction pattern. And from that diffraction pattern, people that are trained in this can determine then the molecular structure of the crystals. So here's the X-ray crystallography diffraction pattern for DNA. That X in the middle tells you it's a helix, and the dark spots on the bottom and the top tell you that the bases are perpendicular to the main axis. So here's Watson and Crick with their model in 1953. That's James Watson on the left, Francis Crick on the right, pointing to their model. And it's thought that Watson went and stole the diffraction pattern from Rosalind Franklin, not knowing what it was, but scribbling it down on a napkin and bringing it to Francis Crick, who was able to deduce it because he'd been trained in that kind of stuff. Watson from the United States and is still alive, very, very elderly though. Crick is deceased. He's an Englishman. Watson was only 23 years old when he was doing this as a postdoc. So at 23, he already had his PhD and was doing Nobel Prize type work. And this is Maurice Wilkins, who shared the Nobel Prize with them in 1962. He was Rosalind Franklin's boss. So the Watson-Crick model says it's a two-stranded double helix, the molecule, with two anti-parallel sugar phosphate backbones. The bases are paired A to T and G to C inside the molecule, and the base pairs are stacked perpendicularly to the axis of the molecule. It's got major and minor grooves, and the nucleotides, the building blocks of the molecule, contain a sugar component, a base, and a phosphate component. And again, for this, Watson and Crick shared the Nobel Prize in 1962 with Maurice Wilkins. So then here's a view of the DNA molecule. PS is sugar phosphate. That's the backbone. It's the same all the way up and down the molecule. And then the you can see that the GC base pairs are held together by 3 hydrogen bonds. The AT base pairs by just 2 hydrogen bonds. We're probably introduced to this in your introductory bio course. Here are the sugars found in DNA on the right and RNA on the left, and the only difference is that the 2' carbon in RNA has an oxygen, and DNA it does not, hence the name deoxyribose. So we can see the sugars are numbered 1' through 5'. 5' and 3' are important in distinguishing the direction that the DNA molecule goes into. The bases are purines and pyrimidines. Purines are double ring structures and pyrimidines are single ring structures. Remember that uracil replaces thymine in RNA. Here's a close-up of the purines, close-up of the pyrimidines. There's a phosphate. So here are a couple of the nucleotides, the purine nucleotides. So we can see that the phosphate is stuck to the 5' carbon, where the bases are stuck to the 1' carbon. And here are the pyrimidine nucleotides for DNA. And then here are the 4 nucleotides shown together. So we talked about the anti-parallel nature, and notice that the 5' to 3' is how we distinguish how the sugar phosphate backbone of the molecule runs. And you notice on the left there for DNA, it's running down in the 5' to 3' direction, and on the right, it's running up. And we see over here on the right, RNA is a single-stranded molecule. So here's the double helix structure of it. We can see the minor and major grooves there. This has them labeled, major grooves and minor grooves. These are important for the attachment of proteins to the molecule. This shows that again. Here's some Drosophila DNA, so several millimeters worth of DNA is coiled up in every cell nucleus in a fruit fly's body. Our genome would stretch out to be about 2.5 meters. That's about, 2 meters, that's about 6 feet. So all that DNA is crammed in every cell nucleus in our body. Often the DNA is methylated, and this is done by enzymes, and A and C are commonly methylated, and this is used to distinguish the native DNA from viral DNA that viruses want to inject into our cells. So here's 5-methylcytosine, a methylated cytosine base. DNA has bends in it. Usually 4 or more AT base pairs causes the DNA helix to bend, and this affects how proteins bind to the DNA. For instance, that sex-determining region of the Y chromosome, the DNA there bends at about 80 degrees so that transcription factors, which are really important proteins for transcription, can bind there. And that's what this shows, a DNA bend here with the ribbon-like structure is the protein. DNA can also form hairpins with a stem and a loop based on the base sequence, of course, or just a stem only, or a double hairpin, which is known as cruciform structure. RNA also has complex secondary structure. Here's a single RNA molecule, and towards the lower left are the 2 ends of the molecule, and you can see they sort of pair up with each other at various points, forming lots of hairpins and large bubble-looking structures as well. In the black box there, we see the base pair sequence of that region of the RNA molecule. Now, all DNA ever found in cells is negatively supercoiled, and that's how I guess it can be wadded up to such a small size to fit in the nuclei and mitochondria and so forth of cells. Topoisomerases are enzymes that do this negative supercoiling. And this shows you, it's sort of like an old-fashioned phone cord. Phones were like this when I was a kid, maybe not when you were kids, but when I was a kid. So anyways, this kind of shows you on the right there, the negative supercoiling, and there's a picture of it just to the left, sort of like a twisted up old-fashioned phone cord. It shows the last two slides together. Now we're getting into the genomes of bacteria and viruses. So E. coli has a genome that's got about 4,000 genes and 4.64 million base pairs. In the cell, it's of course folded and supercoiled to fit in the nucleoid region of the cell. It contains these loops that are independently supercoiled. These are called domains. There's anywhere from 50 to 100 of them. This shows that the open circular chromosome is 350 microns across, but when it's attached to a DNA, an RNA molecule, in showing you its independent domains or loops, it's down just 30 microns across, and then with each supercoiled domain, the chromosome is only 2 microns across. And then towards the right there, it shows you what happens with partial RNA degradation, digestion, or DNA digestion. And so here's our relaxed DNA molecule on the left, and in the lower right there, a supercoiled DNA molecule, how they really occur inside of prokaryotic cells. This shows you the DNA spewing out of the cell. So when it's not supercoiled like that, there's no way it can be all crammed inside the cell like that. Now moving on to viruses, this shows T4 bacteriophage perched on a bacterial cell. So the blue stuff is bacterial cytoplasm, the yellow stuff is outside the cell, and the red thing that looks kind of to me like a lunar module is the phage itself, and it's in the process of injecting its DNA into the bacterial cell. Viral genomes can be RNA or DNA. The smallest viruses tend to have RNA genomes. One has only 3 genes. A virus called MS2 has 4 genes, and its entire genome is only 3,569 base pairs. Smallest DNA virus has 9 to 11 genes. One with 11 genes has a, as you can see there, 5386 base pair genomes. That's pretty small. On the other hand, one of the bacteriophages, T2, has up to 150 genes. And this shows bacteriophage genome map for the bacteriophage Phe-X174, showing you that the genes there are labeled by letters. Some are discrete from each other, but others are overlapping. For instance, gene B is contained within gene A, gene E within gene D. That's kind of interesting, I think. So here are HIV virions in a T cell. So those things that look like little speckles of peppercorns or something in the middle there, those are the virions, and the cell's about to lyse and will be destroyed as it releases the new virions into the body. So the structures of retroviruses are really pretty complex when you think about it. It's not a living entity. We've got a single-stranded RNA genome, 2 copies of it, with the reverse transcriptase there, surrounded by capsid proteins. That's all surrounded by a core shell of proteins, which is then surrounded by a phospholipid bilayer with glycoproteins for attaching to cell surface proteins. Lastly, comparing genome sizes of different organisms, you see viruses are quite small, bacteria a little larger than viruses. Algae run the gambit from being just a little bit larger than bacteria to very, very large, and also some amphibians and fishes have very large genomes, and we don't really know why that is. And so with that, we are done with this.